Okay, so up till now, we've done all of these organelles. And like I said, the basic purpose is to be able to understand what each of these represent, what each of these says. The moment you see a lysosome, those glyoxysome, peroxisome, the secondary vacuoles, everything should just come back into your mind. You should be able to reflect on it. You should be able to reflect on the purpose of each of these. You should be able to look at a mitochondria and be like, okay, I see the way we think inside. It has an F1 particle. I see these dots on the endoplasmic reticulum. It's definitely the rough endoplasmic reticulum. So this is stuff that you need to be able to pick just by looking at this, right? So let's move on to one of the more important topics, right? So that's prokaryotes and eukaryotes. Now, I'm sure you guys have an introduction of it already. If you guys don't, then let me just tell you that there's two kinds of cells. The prokaryote, which was essentially the first one being studied. So the prokaryote, and the word, like the word pro says, it's the initial. So this is what we ideally characterize a prokaryote as. And then we have a eukaryote, which is essentially this cell, which looks a lot like your animal cell. It has, you can see now you can see, look at the nucleus, the nucleolus, everything that we studied up to now. And look at the stacking next to the nucleus, definitely the endoplasmic reticulum. Can't see the ribosomes, so can't really say which one is which. So I'm not going to assume. Then we have another stacking, which is the Golgi, of course, and I can see the trans phase from which vesicles are bleving out, and then I can appreciate the mitochondria. So, and I see a cell membrane, I don't see a cell wall, and its excretion is occurring, so the Golgi is pretty active, right? And then I can see this molecule is entirely different. It has something else going on, right? It has no nucleus, but it has this fibrin-like network, which is essentially the DNA. So that's essentially the pro and the eukaryote, and that's exactly the differences between them. The prokaryote has a cell membrane, of course. It has DNA, but that DNA is not surrounded by a nucleus. So one key thing that you need to understand over here is that the essential difference between pro and eukaryotes is that prokaryotes will not have internal cell membranes. There is no internal cell membrane. So any organelle which had cell membranes will not be present in the prokaryote, right? DNA does not have a cell membrane. It will be present. Ribosomes do not have cell membranes. They will be present. Golgi does have, it has a double cell membrane outer layer, so it can't be present. Endoplasmic reticulum can't be present. Mitochondria can't be present, right? So anything inside the prokaryote cannot have a internal cell membrane of its own. Except the only membranes that are possible are maybe an internal and an external membrane, with the internal membrane being folded upon itself. And those foldings can have enzymes or whatever, but it cannot have organelles with their own independent membrane. So the pro, just remember that. And that makes life so much more easier. You can remember this drawing if possible. Just by it, you can understand that prokaryotes can be motile. They can have these tails called flagella or hair called cilia. Again, if you remember, microtubules were making these. So microtubules can have different functions in and prokaryotes. In eukaryotes, you remember the functions. In prokaryotes, there's an additional function such as movement or motility. But again, even in eukaryotes, there's motility. For example, the sperm cell. Now, the sperm has a flagella as well. So again, there's certain things that are, so what I mean to tell you is that there's certain things that you might look at this diagram or any diagram, because there's like hundreds of things that are different in both of them. So you might think, okay, so I've seen a flagella on a prokaryote. So the question comes up, do eukaryote cells have flagellas? And you completely miss the fact that sperm does have a flagella, right? So I want you to not do that. I want you to understand that the essential difference is the difference in meaning. And the difference in meaning is just simple. No internal cell membranes. And that's how we're going to go ahead with it. So any difference that we have, it's going to be on that. For example, eukaryotes will have a nucleus. Prokaryotes will not. The ribosomes, like I said, prokaryotes are much smaller in size because they don't have internal organelles. They're that larger ones, so they're much smaller. Eukaryotes are much larger. Prokaryotes usually exist singular because they don't have internal organelles, so they can't support a whole organ system. They can only support themselves. So, for example, bacteria, now mitochondria, they all exist singular in nature. None of them exist like us. We exist at a multi-organ and a multicellular level. Our cells are dependent and codependent, and we form tissues such as the blood and the brain and whatever, everything's going on in there. Ribosomes, prokaryote, the size is smaller. The ribosomes size is smaller. Prokaryotes are 70s. Eukaryotes are 80s. Again, while answering your questions, be specific about them. You should know which one is which. The endoplasmic reticulum, Golgi, mitochondria, anything, lysosome, vacuole, anything with internal membranes is absent in the prokaryote. So make sure you understand that as well. Then eukaryotes and prokaryotes both may or may not have a cell wall, right? Some prokaryotes may have a capsule, but most of them end up having a cell wall. Not all eukaryotes have a cell wall. Some do. Animals don't. Fungi and plants, they do, right? So you need to understand that the membrane and membrane-bound organelles are the defining feature. The examples are very clear. There's bacteria and archaea that are prokaryotic. Archaea are not in detail in your syllabus, so they won't be tested. Bacteria will. So bacteria is important. It's important to know bacteria as an example of prokaryotes. Other than that, everything else is eukaryotic. There may be certain differences between these eukaryotic cells within themselves, which we'll study about as well. Again, the cell size is smaller, nucleus is, there's DNA, but there's no nucleus, DNA replication. And so this is one important thing to know. DNA replication, because in a DNA cell, it's bound in, it's occurring in the nucleus. So they can start from any corner and it can start simultaneously, right? It can start occurring in any corner of the DNA. But in prokaryotic cells, because it's open and it's so exposed to the environment, only one area opens and DNA replication starts at that area and then completes. There's no multiple areas where DNA replication starts and ends, right? Remember, DNA replication makes DNA forks. So always remember that in prokaryotes, only it starts from one area and extends to complete itself. It never occurs at multiple areas in the same time, okay? Again, unicellular chromosomes, so chromosome, because there's one DNA and that DNA is generally, so that DNA is in a single loop. So it's a circular DNA. It's not linear. Eukaryotic DNA is linear DNA. And then there's an additional thing called plasmids in the bacteria. Plasmids are a single loop of DNA that can jump from one bacteria to another. Our cells cannot do that. Our DNA is bound to the nucleus. It can't jump from my cell to your cell, right? But bacteria A can transfer its DNA into bacteria B by a circular thing known as a plasmid. Again, growth is obviously faster when your organ is so simplistic. Organelles are obviously absent, and then they have DNA, so there's hereditary component present, and the cell wall. So cell wall, remember I showed you bacteria cell wall. You can still Google it, Google gram-positive and negative bacteria. Remember how difficult and complicated it was? There was peptidoglycans in some places, others in not having it. So all of that is quite complex. So it's present, and we divide bacteria. So remember this, we divide bacteria into two types by presence of a different cell wall, okay? So that was prokaryote and eukaryote. This is a simplistic diagram. Again, like I said, this diagram is important because you need to be able to understand that there are some structures that will always be present. For example, you will always have 70s ribosome. You will always have a peptidoglycan cell wall. You will always have a cell membrane. You will always have a circular DNA, not a linear one, a circular one. You can have a flagella in both eukaryotes and prokaryotes, but more in understand that in prokaryotes, if these are some can haves, not must haves. A slime capsule can be present, and then an infolding, like I said, an internal membrane infolding known as a mesosome. So a mesosome can be present. This is called a mesosome. This can have enzymes, pigments, whatever is needed. And then there's plasmid. So it's a very small bud off from this normal DNA. And this can be transferred from one cell to another cell. And a special organ that transfers this plasmid from one bacteria to another is called the pili. So pili are sexual reproductive organs. What does that mean? It means it's going to stick to the other pili of the other bacteria and just transfer this plasmid across. Okay, so the transfer of the plasmid is what is essentially happening. Then we need to understand differences between a plant and an animal cell. Again, it's very easy. They're both eukaryotes. So ATS ribosomes. So if I ask you for similarities, just use those words, ATS ribosomes, and this and that. All of those that we studied were different from prokaryotes, right? And then there's some similar stuff. So similar stuff is easy to do. What's interesting is the different stuff, which is in different colors over here. For example, animal cells do not have centrioles. We do not have essentially. So lysosome has been used as a very specific term here, but one thing I need you to focus on is centrioles. Animal cells do not have centrioles. They have another system of control that is far more complex. They have another system of forming the spindle, which is far more complex, right? And then the animal cell will have a cell wall, and it's going to have chloroplasts in addition to the mitochondria, not an exception to it. Remember, both will be present in the cell. However, there are no plastids in an animal cell. They're only in plants. The specific plastid can be chloroplast. In petals, the plastids can be colored ones. In fungi, we can have plastids as well, which are not necessarily chloroplast. In algae, for example, they can be chloroplast as well. And then there is something called plasmodesmata, which are gaps between two adjacent cells. So this is a better diagram explaining it between two adjacent cells. So you can look at it and without even seeing this right here, you can tell it's a plant cell. Why? Central large vacuole, chloroplast, nucleus pushed to the side. This you can imagine this is the Golgi, and this is the you can imagine this is the endoplasmic reticulums, right? And then looking at this cell, between these two cells, if there are holes present, these holes are called plasmodesma. A couple of them are called plasmodesmata. So that's the important stuff here. Remember, plasmodesmata, a couple of holes between plant cells that are not present in animal cells, right? This is another real view, right? It's real because it's not colored. It's through the electron microscope to show you that you're not supposed to get confused. Look at this. The vacuole can be split into two. Again, your general idea is supposed to be to look for chloroplasts, to look for a large vacuole, which can be split into two, because what is a vacuole? We have smaller vacuoles in cells, but in plants we have a large vacuole that can be split into two with the nucleus in the center. Can you appreciate this is an electron microscope diagram? And because they expect you to know electron microscope diagram in A level, so at any point they can decide to put something similar to it up in an MCAT exam. So just make sure you understand that when you're looking at it, you're not getting confused. You're understanding that, okay, this looks like a vacuole, this looks like a nucleus. Only one thing has such a large vacuole, a plant cell. This looks like a different structure. So if I can't really tell what it is, I can imagine it's a chloroplast. It looks different. It looks like stackings. Remember those stackings of the point-like structures of the thylakoid membrane? It looks like that, right? And then and then it's making food. How do I know that? Because look at this granule. The starch grain is being produced here. So remember the function of plastids and chloroplasts, right? Okay, so the next topic is understanding our cell membrane, right? And in understanding the cell membrane, something that's very important is understanding the behavior of a molecule that we essentially call a phospholipid. So essentially what happens is you generally have a fat molecule, right? A fat molecule is something that looks like this. So a fat molecule has a body and three tails of fatty acids. When you remove, and it's essentially hydrophobic. So there's two terms, it's hydrophobic and hydrophobic and hydrophilic, right? So hydrophilic means loving, philic means loving, and phobic comes from the word phobia, which means you're scared of it. Now, a fat molecule is hydrophobic, so it's scared of water. It's going to run away from water. If you put water molecules in this direction, the fat is going to end up on this side of the screen. It hates water so much, right? A hydrophilic molecule will end up here because it loves water so much. Not This is not a fat, it's some other hydrophilic molecule. Now imagine I remove this one tail and I keep these two tails and I replace this one tail by a phosphate. Now a phosphate molecule is hydrophilic. So now you have a special kind of a molecule. It looks so awkward, like a little cheap, but I'm going to improve how it looks just by making it this way. So I'm making its philic portion at the top as a head, and I'm making its two tails, and this is the body. It's on the inside. I'm not showing the body. I'm just showing you that there is a molecule which is called a phospholipid. The phospholipid has a philic portion, which loves water, and a water-hating phobic portion, right? So the phobic portion, now, if I put many of these inside the water, the inside of the water will face the loving portion, and automatically the phobic portion will end up on top, moving away from the water. So the philic portion ends up in the water, and this is how it looks. And now imagine, so let me clear all of this and give you a better perspective. So now imagine I'm putting many of these, and the ones on the inside, they're all traveling to form balls like these. Now they form balls like these. Why? Because they want to protect their hydrophobic portions. So they're smart enough, they're smarter than you and I, obviously, because they're made up of inorganic molecules, right? So they're smart enough, the ions detect that, okay, my water loving portion is over here. Why not I face all of my water loving portion on the outside and the water hating portion, hydrophobic portion, on the inside? This way I can protect this and cover the outside. And the heads form a impermeable membrane. It does not let water enter, right? Now, now, it's very interesting. Remember, whenever you're talking about membranes, you're saying they're semi-permeable, which means they're not letting water in. So that's essentially what a membrane is. It's essentially these phospholipids, they're so clever that they arrange themselves in this way. Their tails, so they form a bilayer. They're so smart, they form a bilayer. Instead of forming this closed up sphere, they form a bilayer in which they're comfortably arranged with the heads pointing outwards and the heads pointing inwards of the second layer facing the water, and the tails are interlinked with each other, not letting anything through except for a few things, right? So they're selectively permeable, and that's exactly how to just define your cell membrane. So you should be able to understand this very well. If you still cannot, feel free to send in your questions. This is a complicated topic. It needs to be understood once, and if it is understood well, it sticks with you forever. Now imagine this as a 3D sphere. That this automatically becomes a cell. The outside is the water and the inside is the cell. Now if you put in a nucleus, if you put in organelles, this is automatically the inside of a cell. So that's exactly what ends up happening. But this part is easy to understand. This is where you need to put in your brain, right? So you remember that molecule. So remember that phosphate and the lipid tail or the fatty acid tail, right? So the fatty acid tails can be straight or they can be kinked or whatever the shape might be. But you need to understand that structure of this membrane. This is a membrane. It's a cell membrane. This membrane can be a cell surface membrane on the surface of cells or inside the cell. Remember those vacuoles, even the nuclear envelope. What was the nuclear envelope made up of? It was a similar membrane, similar spheres forming inside the cell membrane. And then they eventually had organelles inside them, right? Or nuclear, if this has DNA inside of it, it becomes nucleus, right? So what is the structure? Is it only phospholipids? No. It can have proteins, large proteins called integral. Integral means extending from one end of the membrane to the other end, or peripheral, which means in the outer layer only. Or they can just be attached outside, which means they don't disturb the membrane, they just lie outside. It can also have cholesterol in the middle. Cholesterol acts similar to phospholipids. It attaches itself similarly in lines. So what does the fluid mosaic model tell us? It tells us that essentially there's a bilayer of phospholipids that are fluid. They are continuously moving. Like I told you, there's no bonding, nothing at all, just those hydrophilic and hydrophobic attractions. And within them are proteins, right? So there's a mosaic, there's a random arrangement. What's a mosaic? It's a random arrangement of stuff, right? It's a mosaic of protein and phospholipids, and it's fluid. It's continuously moving, right? So now we need to understand the function of each of these. If you understand what each of this is made of. So we have a phospholipid, which is fluid, like I said, and it controls what goes in and out, makes it semi-permeable. Then we have cholesterol. Now, cholesterol is very important in controlling fluidity. Remember, cholesterol arranged itself in next to these phospholipids. If you increase or decrease the concentration of cholesterol over here, for example, if you remove half of these purple ones and make them yellow, they will be much more fluid, because now they can't be held together as strongly. We'll talk about this more as well. But then there's proteins. Proteins can be to anchor cells. For example, cytoskeleton can attach here to anchor different organelles, or anchor or from the outside peripheral protein, it can anchor itself to the next cell. So it can be different reasons. They can be channels, because for example, large ions cannot pass through. Remember, I told you these are water hating. So a large ion like calcium cannot pass through something that is already hating ionic substances, right? So if you, for example, imagine you make this protein hollow, you make a hole in it, now anything can pass through it. So these proteins can form channels, they can form gates. These gates can open and close for specific proteins or specific molecules, specific hormones, right? And then there's other molecules called glycolipids or glycoproteins, and that's very easy to understand. So I forgot to put in a picture of it, but I'm going to draw something for you, which is going to make it so much simple. So a glycolipid is just this lipid molecule being attached with a glucose chain, right? It can be kinked up, it can be in different ways. So this is called a glycolipid. And if that glucose chain is attached to this protein, then it's called a glycoprotein, as simple as that, right? Or any protein, even if it's this, or even if it's this. Now, this is again a lipid molecule. Cholesterol or sterols are lipids. So this makes it a glycolipid, right? As simple as that, right? So now we need to understand that glycolipids and proteins, what do they do? They're like the sticky arms. They're like the handshake arms. What do your arms do? They handshake with the other cells. They signal other cells that I'm here, don't push me. They're like receptors. They can receive signals. They can receive hormones. They can receive a hormone, and the hormone, if I hand you a piece of ball, you can understand, okay, now I have to throw it. Just like if you show a dog a treat, he knows he has to eat it. So the similar way, they're like your hands. They detect stuff, they meet stuff, they hang on to stuff. So that's everything they do, right? So it's again very simple, just putting it here so you know that it's an important. So when you go through your books, you know that it's an important part of your reading material, right? Again, so very easy. You have, whenever you have a ligand can be anything: a hormone, an enzyme, a receptor, a stimulator. These receptors are those glycolipids and glycoproteins, and when they get activated, the cell performs a response. For example, changes its color, changes its speed of activity. When you're not all cells will have, which is why I did not show you glycoproteins and lipids over here, because not all cells will have them. Why? Some cells may have more, some will have less. If the other cell, which is not a target, does not have the same glycolipid or glycoprotein, even if the ligand comes here, it won't be able to detect it, and this cell will do its normal functioning. The functioning will not change. So this is how the cell varies, different cells vary, how they respond to different signals. Now, again, just to show you how small that cell membrane is, it looks like two dark lines, even if you zoom in by 250,000 magnification, right? Phospholipids are so small. And then you need to understand what affects their fluidity. And it's very much common sense. So temperature. Temperature freezes everything. So it makes things non-fluid. Heat speeds up everything, right? It makes it more fluid. And because they're not held together by gluing or bonding, so the temperature will affect them, right? Cholesterol, like I said, too much cholesterol means that now in one layer, if there's too much, if this layer has one cholesterol and this has two, phospholipids can be tightly packed together, but cholesterol is this kinky molecule. It disrupts that packing material, so this makes this layer more fluid than the layer on top. Now, and that's how the cell controls your body. For example, if it gets too cold, what happens? All of your cells start making more cholesterol, right? Why do you eat all of those nuts and everything and omega-3 fats and all those fatty stuff in winters? Why? You're not bears, you're not storing it to go to sleep. What you're doing is you're providing your body with extra cholesterol just to make sure that your body, your cells can now use it to, because cold, remember, cold will make it very rigid. You don't want it to be rigid. You want it, it will break otherwise, right? So what you do is make more cholesterol. As a result, increase the fluidity, right? Similarly, if it's too hot, what do? You remove all the cholesterol. You remove all the fluidity. You make sure that the integrity holds. Otherwise, the shape won't hold. Okay, and then so essentially it acts like a buffer. What does a buffer mean? It doesn't let too cold or too hot temperatures affect the cell's integrity, right? And then there's nature of those fatty acids themselves. So it's always important to understand, whenever you're talking about something, you need to understand that things are external and internal. So we've talked about an external factor, temperature, we've talked about an internal factor, cholesterol, and now a self-internal factor for the fatty acid. So if what's the difference between a saturated and unsaturated fatty acid? A saturated fatty acid has carbon linked to carbon that does not have a double bond. An unsaturated one has a double bond. Unsaturated essentially means, so saturated means fully occupied, right? If carbon does not have double bond, it is fully occupied. If it has a double bond, it's not. Each double bond will appear as a kink in the chain. So these are kinked chains. And as you can imagine, unsaturated or those with double bonds are kinky, and kinky ones do not align as neatly. So, pun intended, you can understand it for your own self, right? Okay, so then we need to understand the substance and what passes and what does not pass. So lipids and fats, if they're neutral, obviously lipids and fats are neutral, or anything that's neutral, it's easier for it to pass because you can understand hydrophobic and hydrophilic properties are not being affected, and smaller things will obviously pass easily as compared to bigger things. Again, smaller molecules that are non-charged will pass. But what if they're charged? Look at this. So we talked about lipids and fats generally, which are uncharged molecules. They do pass, right? But smaller pass more than big. But what if we have charged molecules? So if we have charged molecules, smaller molecules might be able to pass, but larger molecules, they will never pass. Large ions or anything. As such, for example, hydrogen ion, you know, H positive only has a nucleon and no electrons, so it can pass, obviously pass, right? It's because even if it's charged, it's so small. But if there's a large molecule like magnesium or potassium, those or chloride, they can't pass. They're larger and then they're also charged on top of that. So what do they need? Protein channels. So study more about those protein channels. They're very easy. They can either be channels or they can be gated ones, right? So very simplistic. And then finally, we have types of transport. So types of transports are very simple. There's either active transport mechanisms, active transport meaning you need to use energy. And if you understand you need to use energy, you will also understand that energy is very important for the cell. Energy is in the form of ATPs. Remember, mitochondria makes them. It's so important that the cell does not want to waste it, right? So the cell will only use energy when it's transporting things that it does not have. That's essential to understand. For example, because you know the basics, now you need to get the concepts for the MCAT. For example, you have a high amount of substances outside the cell, but a very low amount inside. Now, that low amount inside, you're deprived of substances, you're hungry. The cell need, sorry, a very large amount of contents inside and a very low amount outside, right? So now you're hungry, you're deprived, you need those contents because they're quickly being used up. And even if they're not, you're so scared, you're selfish. You're like, okay, if I don't, you get all of these three inside right now, somebody else will take them. I need them. Now these are essential to you. So now you spend energy to pull them in. And that spending of energy to pull something in is known as active transport. You always need a pump for it, a suction, imagine a suction pump. You always need a suction pump for it. That is the protein. And then always remember, like I said, the concentration outside will be so limited, it will be so rare, you will need it, which is why you're spending that energy on that pump, right? So what you're doing is you're moving something from a low concentration to a high concentration because you want it so badly, so you're spending energy on the pump, right? So that's essentially active transport. Other mechanisms are passive ones. You don't need to essentially spend energy. So for example, diffusion, it's just the movement from area of high concentration to low. It's just like mixing. If you put a red ball in your bathtub, all of it will become red soon. Why? Because it's mixing out. It's going from area where it's high to an area where it's low, right? Another passive process that can occur is facilitated diffusion. And like the word says, it's facilitated, which means something is helping it move. For example, if this is magnesium ions, they can't move across the cell membrane, they need a facilitation. This protein channel, remember that protein? It's now this has a tunnel in it. This protein channel will become a facilitation. And so if there's a movement without energy from high to low, through a protein channel, it's known as facilitated diffusion. So again, there's diffusion that simply, it can be across a membrane. If it's water across a membrane, that's known as osmosis. So remember that. Diffusion is a general term used for anything, gases, solid, liquids. If it's specifically water across a membrane, water and membrane, two key terms, then it's osmosis. It's not diffusion any longer. Facilitated can either be through gated channels or a tunnel-like channel, right? And then there's active transport, which is using ATP and turning in from low concentrations to higher concentrations, okay? So very easy stuff. And then the last process is endo and exocytosis. Remember those vacuoles? Did none of you ever wonder how they form? It's very simple. What happens is this cell membrane blebs inward to surround all of that material. Imagine if this was outside the cell, then it's endocytosing something. If it's solid, then it's endocytosis. If it's liquid, it's called penocytosis. Similarly, if it was forming a vesicle, that can now be digested by lysosome or sent anywhere where it's needed. And if it's going backwards towards the cell membrane, it's called exocytosis, right? Very simple stuff again. Okay, so now we have this again, another daunting image, but very important because I want to make sure you're exposed to images from a different perspective, not just a very limited perspective of your textbook, right? So this is an electron microscope. This is a eukaryotic animal cell. So why is he using two terms? He's saying eukaryotic because ATS ribosomes, nucleus, not a bacteria. And then the specific kind of, it's an animal cell, it's an electron microscope. You can appreciate there's a nucleus, and other than that, you just need to be able to look at it and not get scared. And if there is a question from it, it's going to be relatively easy, hopefully. So that is it. That's our topic for, that's everything for this topic. All questions are welcome.